have, today is Tuesday, correct? The last day of March. It's exciting. And then we have Thursday, and then we have Tuesday, and then we have Thursday, and this is going to be our third exam. I heard the sound. Sweet carbonation, meeting the world. So there's our exam 3. So we have 3 days. We're going to hopefully spend most of Tuesday reviewing, as is our custom. We may have some loose ends to tie up. The review is posted if you haven't looked at it yet. We've completed photosynthesis, which was a big section, but not the whole thing. We're going to talk about cell signaling next. I think I'm going to go in the order on the sheet. Maybe that will make your organization a little easier when you study. Cell signaling is actually kind of 2 little side topics that we're going to hit today. And then we'll do mitosis and meiosis, which I think you probably have some history with. I'm going to guess if you've been in any biology at any time, anywhere in your life, you had to have talked about cell division, right? That's just one of the go-to topics. So hopefully that will be kind of a walk down memory lane. And then, excuse me, after we do the cell cycle, the next natural thing will be to talk about cancer and how the regulation of mitosis is intertwined with cancer, and maybe learn some new stuff about that, okay? So that's where we're at. I do want to hit a couple of the highlights in photosynthesis. If by chance you did not receive one of these, if you happened to miss last week, before you leave today, grab one of these sheets and beg one of your classmates to give you the answers to fill that in. I'm sure parts of that are going to be on the exam, without a doubt. And I'm sure because I wrote the exam. All right, so let's spend just a couple minutes and hit these highlights. I don't want too much time to pass before this slips out of your mind. So let's just do the couple minute overhaul here. We're not going to pause or go into detail. We're just going to hit the highlights. All right, so in chapter 8, we talked about the cycle of organic molecules and carbon dioxide and water. So we produce carbon dioxide when we break things down like sugar. You probably recognize the chemical formula for glucose. And we need oxygen to do that. In the process, we build up a gas that we cannot use, but plants can. So plants use the carbon dioxide to produce sugar precursors as well as oxygen. Okay, this is located inside, of course, plant cells, and those plant cells have chloroplast. The gas exchange to get the CO2 and to allow the O2 to leave is through spaces called stomata. Those are openings that get gas into the cells, the mesophyll cells, or mesophyll, some people say, where this is taking place. Within those cells are chloroplasts. You'll want to know a little bit of this anatomy, and there is just a little bit, not too much to know. Namely that within the inner membrane, all right, so we've got a double membrane just like mitochondria have. There's DNA inside here. It's not shown in this diagram, but there is, which just makes it unique among organelles. Mitochondria and chloroplasts both have their own DNA. Inside the chloroplast inner membrane is the stroma. This is the fluid within which the granum exists. And granum is a singular of grana, which is the plural of these stacks of discs. The discs are called thylakoids. Inside the thylakoid, just like inside any membrane-bound compartment, is the lumen. Right? Lumen just means the inside of a membrane-bound compartment. Okay, if we then think about our reactions and where they act, a question I can imagine myself asking is, which compartment in the thylakoid do the light reactions occur? And you're going to choose the thylakoids, because it has to be the thylakoids because that's where the pigments are. Okay, you can't have light reactions without pigments because the light is absorbed by the pigments, and that's where the energy gets extracted. So the light reactions happen over here. Outside of the thylakoid and the stroma is the Calvin cycle. This does not require light. This can happen day or night, darkness or in light. It doesn't matter. The Calvin cycle is where we're going to take carbon dioxide and produce eventually sugars. We're going to need a ton of energy from that. The energy comes from the sun, which causes the light reactions to work, which produces ATP. Okay? In the process, we need electrons. We're going to get those electrons from water. Water, when it releases electrons, is going to become hydrogen ions and oxygen. And that oxygen then we can use, right? That's where it comes from. Okay, let's keep moving. You're going to want to be comfortable with these concepts. Hopefully your lab shed some light, or science joke, on this, and I guess you may be doing this now to turn in this week perhaps, because you got the data last week. But the action and absorption spectra are looking at the places within the visible light wavelengths where light is absorbed. So in the violet and blue wavelengths of light, 400 to say 475 nanometers, you absorb in the chlorophylls, right? So the chlorophylls are taking those wavelengths of light out of commission, and then they're also absorbing over here in red, and anything they don't absorb bounces back to your eyes. So when you see something that's green, it's because that green thing has absorbed violet, blue, yellow, and red, right? And it's reflected green back. If you're looking at something and it's red, it might be something like beta carotene that's absorbed violet, blue, and green, but reflects back the yellow and red. Actually, it would be orange. Okay, anytime absorption is happening then, photosynthesis can happen. All right, so this can be measured at different wavelengths. You can flash wavelengths of light at a plant and ask, is that plant producing O2? Is that plant consuming CO2? And if it is, then you translate that to the relative rate of photosynthesis. So you don't have like a dial on a screen that says photosynthesis is happening, you know. Instead, it's saying O2 is being produced and CO2 is being consumed. Okay, so those are all important kind of big picture facts. Then we got into the smaller picture facts, which are the places an electron can go in the thylakoid membrane. So this is based kind of loosely on the worksheet and the so-called Z scheme, where electrons who begin their journey in water and then are extracted, and then P680 is the pigment that uses those electrons and raises them to a higher energy level. P680, however, has to give those electrons away. And now we start our journey of electron carriers to pheophyte, to QA, to QB. These things are passing that electron like a hot potato. QB says, I'm getting out of that photosystem, and it takes its electron to the cytochrome complex. Cytochrome complex is a different kind of machine. The cytochrome complex is not an electron carrier. It's going to use that electron to do work. The work it's going to do is pumping hydrogen ions into the lumen. Okay, so that's doing that using energy from the electron. Eventually, the electrons had enough of that, and it travels out to another electron carrier called plasticinin, which takes it to another photosystem where it gets another boost of energy from light, and then that electron has has more energy to share. It gives itself to ferredoxin, which gives itself to NADP plus reductase, and that electron finds its final resting place in NADP plus, which then forms NADPH. Okay, so it's going to do a lot of stuff before it gets to the end. The purpose of all this is to create a hydrogen ion gradient so that hydrogen ions can flow out of ATP synthase, and as they leave through this revolving door, that energy is used to make ATP. Okay? Now we said there's one little sideshow here. This is the thing if you missed last week, you're going to want to make sure that you get from a classmate. If you don't have a classmate you trust, actually this is probably on the Blackboard PowerPoint, is it? Anyone download the images? Okay, so you actually have this. So forget about your classmates. Don't do that. We're all in this together. Okay, one little side thing we said was that sometimes the electrons don't take this linear path to NADP plus reductase. Sometimes electrons take a little side journey where when they get to P700 and they get that energy, ferredoxin says, instead of going forward, let's do another round. And it gives its electron back to QB, and it takes another swipe at this. And the reason it can do this is sometimes you need more ATP than you have. So if you take the original journey all the way to the end, you do generate ATP, but if you stop and do a few loops of this, you make more hydrogen ion gradients and you make more ATP. Sometimes you need ATP more than you need NADPH. Okay, so that's why it has this option. Okay, then we talked about, I'm going to put all this up together, the unique events that happen inside the photosystem 2. And this is where P680 is going to take energy from light, charge up its electron, photoexcite its electron, and then give that energy away. And so the process involves light hitting a pigment, and then when that pigment gets excited, it shares its excitement with all of the pigments around it. So all these other pigments aren't getting light energy, they're getting energy from that first pigment. right? It's so excited. It's just like your infectious excitement when you leave this class and go back to your dorm or your apartment or your home and you share all the exciting things you learned in class. It's basically exactly that happening, only at a molecular level. And so that resonance transfer, as it's called, bounces that energy all around, and then eventually it gets to P680. P680 is a main player in this process. P680 is cycling between three stages, as you may remember. P680, when it's excited, becomes P680 plus, or star, sorry, not plus. P680 star gives away an electron, becomes P680 plus, and then it needs an electron from water to get back where it started. So when P680 star is excited, and it gives its electron to the primary electron acceptor, which I wish they would just write it in there. I guess I have the power to do that. I should just do that. That's pheophyton, right? That's the first movement in our electron transport. P680 then, it's hard to tell in that diagram, but it becomes P680 plus because it's given an electron away. When you give an electron away, you gain a positive charge. And then we need to get that electron back, and that electron comes from water. Okay, so when water is split down here, the electron goes to P680 and he's back in business. All right? So that's a little bit of insight into, that's the only part of this path where we really look at a close, fine detail. All right, and then let's see, there's the Z scheme, and then we switch to the Calvin cycle, so we'll finish our little review out here with the Calvin cycle. Remember, there are three main phases. The first one is carbon fixation, which is where this beautiful enzyme, ribulose bisphosphate carboxylase oxygenase, 4 words we will never use again. We will henceforth just call him Rubisco. It's a great nickname. So Rubisco is the enzyme that is catalyzing the input of CO2 to produce 3-phosphoglycerate. And that 3-phosphoglycerate is then going to go into the second phase. So this takes this carbon dioxide gas and converts it to a solid. Okay? The second phase is called reduction in carbohydrate production because using a ton of energy, it's going to make precursors to the things that we want, glucose and other sugars. And then we have to get back to where we start. And so if we started with RUBP, that was our input, along with CO2, we have to regenerate that. So we get back to that, and then we can start again. So this is an endless cycle called the Calvin cycle. It is the main event of carbon fixation catalyzed by Rubisco. We didn't talk about the enzymes that are catalyzing these steps, and we won't. But if you take cell biology or botany or biochemistry, you likely will. Okay, and we did that, and we're good. That's our 7-minute review, 8-minute review maybe. I don't know how long it took. Too long. All right, how do you feel about that? Medium? Oh, a thumb up. That's like medium well. Just a little bit of pink. Okay. All right, it's a lot. I wouldn't expect you to understand all of that at this point. We still have plenty of time to practice that and learn that, but I wanted to send some refreshers. Okay, so if I follow the review, cell signaling and then apoptosis. So the question is, where is that cell? It may be too soon to do cell signaling. I guess it is. Okay. But I do want to do apoptosis because it feels like leading with the topic of cell death before we go into cell division feels right. Death feels right. All right. I need to take the breath after my photosynthesis sprint. Running on lactic acid. All right, any questions about photosynthesis before we move on? Any questions about anything? Who has cells that are ready to die? Yes, we all do. It's happening. It's healthy. If your cells didn't die, they'd be old and stale and gross. You have to regenerate. Imagine if your skin cells never died and regenerated. Oh, you'd look horrible. You'd be filthy. In fact, most dust apparently is shed skin cells, so there's something gross to think about. Okay, so how do cells die? So this process, if you've heard of it before, you may have heard it called different things. So the real scientific name is apoptosis. Okay, apoptosis is often called in a more casual way cellular suicide. Remember to use that invisible zone there. And cellular suicide was a common way to describe this process when it was when the details were first really elucidated, which would have been in the early to mid 90s. So when I was a young lad in graduate school, this stuff was all very fresh. People were really just starting to understand this. So this, in biological years, is a pretty young topic, which is cool because it means there's still lots to learn. But cellular suicide was one way it was described. Another way it's described, which is probably my preference, and the one I'll use the most, is programmed cell death. The reason I like this depiction of it, which is not my depiction, it's just a very common way people call it, but it really describes what's happening. So cellular suicide sounds like a cell is dying, right? But it doesn't really describe how it's dying. And programmed cell death implies that this is part of a deliberate event. Okay, so programmed cell death or apoptosis or cellular suicide, all these things are referring to a cell dying through a deliberate, meaning not a mistake, enzymatic process. Okay, so there are many ways to die, as you know. This down so it's not blocking your vision there. There are many ways to die. So your cells can die, I'll put this, ... In contrast to necrotic cell death, that's an R, necrotic cell death. So necrotic cell death means death from an injury, well, cell death, not total death. Cell death from an injury or a pathogen. So bacteria could cause your cells to lyse and die. A virus could cause your cells to lyse and die. A mechanical injury, such as scraping your knee on the concrete, that could cause cells to lyse and die. So trauma could cause cells to lyse and die, car accident, something like that. So those things are not your cells deliberately saying something's wrong, we should implode to reduce the impact on our host. This is just saying cells are dying. Okay, so programmed cell death, the point of it would be to avoid inflammation, or to avoid, I'll say it this way. The purpose is to avoid further inflammation or, more generally, we could just say problems for the host. So if your cell has something wrong with it, for example, if it's precancerous, it is better for your body, for your host, if that cell were to die than if we're allowed to divide and keep living. Okay, so apoptosis takes care of these cells that are not going to do your body any good, but if we can get rid of them, maybe that will, be for the benefit, the bigger benefit. Okay, so that being said, let's look at the process, which is now pretty well understood. Another place apoptosis works, while I'm thinking about it, is during development. So you're born with more cells, I shouldn't say you're born. You have more cells during development than you're going to end up with. So one example, if you can remember back to when you were a fetus, you might not remember that, but you might, who knows. At one point you had cool little webbed hands, right? While you were swimming around in your mommy's amniotic fluid, your little T-Rex arms, you had webbed feet and webbed hands. And these cells for most people, disappear before you're born. Some people do have webbed hands. It's not, I mean, it happens. It's an exceptional case, but it happens. And so apoptosis is an example of these, or here's an example of apoptosis. These cells undergo apoptosis because you do not live in water. Now, if you were a little froglet or some amphibian or something that needed webbed hands, of course, you would keep those. But all organisms that develop webbed hands during embryogenesis and fetal life are not going to keep those. And so there's an example where apoptosis is not because of an injury or an infection or some sort of immune issue. It's simply part of your normal development. Okay, so how does it work? So we begin, and again, this is a little bit out of order from the rest of the chapters we've been doing, but since we're talking about the cell cycle, I always feel like this is the appropriate time to say it. So you can just use the figures that I have in my Blackboard slides if you want, and look back and cross-reference with your book later if you like. But it begins on the outside surface of the cell. So this, by now, I hope, is a familiar concept to you, which is a transmembrane protein, right? It's a membrane protein that is embedded in the phospholipid bilayer and extends into the extracellular side as well as the intracellular side. And this particular type of receptor is called a death receptor, which means that it's going to receive a signal to tell the cell to die. All right, and that death receptor is going to receive a signal from a signaling molecule. So I'm going to give you an example of a death receptor. So we have real names for this, so we don't just vaguely say death receptor. An example of a death receptor is something called the Fas receptor. Okay, and the Fas receptor binds to a signaling molecule. So an example of a signaling molecule is something called the Fas ligand. Now this is a word that I don't know if we've used it yet or not for you. Say that word in so many different classes, I forget who we've talked about it with and who we haven't. But the Fas ligand binds to the Fas receptor, and they have this perfect match, right? It's just like when a substrate binds an enzyme, it's a perfect match. That substrate was meant for that enzyme. Their shapes interact, their pieces interact, their charges interact. It's perfect. The same thing is true for signaling molecules and receptors. The idea of a ligand is a general term, just like receptor is a general term. So let's define that, because this word will follow you throughout your biology days. A ligand is a molecule that binds to a receptor. Very general term. So if you have a receptor, there's going to be a ligand that binds it, or it wouldn't be a receptor. So the Fas receptor has the Fas ligand. There's a death receptor called the tumor necrosis factor receptor, which binds the tumor necrosis factor. The enzymes, maybe we've mentioned them before, that cause, or the receptors that cause you to produce different forms of melanin are called melanocortin receptors. They have ligands, which are melanocyte-stimulating hormones. So anytime there's a receptor, there's a ligand. In this case, the death receptor has a signaling molecule, and I think does it even say ligand? It doesn't. I wish they would say ligand instead of signaling molecule, because no one calls it signaling molecule. In any case, there it is. So our death receptor binds the signaling molecule, which is the ligand, and this is a stepwise event. So we're moving to the right, but we're looking at the same proteins. So this guy binds the ligand, and then it bundles up with two other receptors like it. Okay, this is a common theme in cell receptors, where you have multiple individual pieces that when the ligand comes along, they all cluster together. All right? And when they cluster together, they start to behave differently. And when they're clustered together, they then produce this thing which is called a death-inducing signaling complex. What a grand name. And the death-inducing signaling complex has the effect of bringing this protein, which is called an initiator caspase, into contact with the death receptor. Okay, so let me maybe spell out some of the sequence of events. So this will make sense later when you look at it. Okay, so the sequence of events, let's just start a list. The first one is a ligand binds a death receptor. Okay, and remember when the ligand binds the death receptor, multiple subunits come together. It's like the Beatles song. So the ligand binds the death receptor. Let's just spell it out. Causing the aggregation of 3 death receptors. Aggregation means the same thing it means outside of scientific circles. It means things are coming together, aggregating. So when these 3 death receptors aggregate, so this is actually accurate, this isn't sort of symbolic, it becomes a trimer, a 3-part structure. When that happens, let's see, say it this way. Initiator caspases can now associate, which means interact, with activated death receptors. Caspases are one of the most important enzymes involved in this process. So you already know it's an enzyme because it ends in ase, right? The question is, what does a caspase do? And so let's have a little sidebar here to define caspases. Caspases are proteolytic enzymes. So if you had to guess, well, you don't have to totally guess because you might see some word roots in here that you recognize. Proteolytic enzymes. Anyone want to take a stab at what that might mean? To be a proteolytic enzyme? Anyone want to ring diamond earrings? Got to ask what happens in here after hours that people are shedding earrings. I just want to know.
You think it's related to the coffee?
It might be related to the trampling of the DNA molecule.
Do they break down proteins?
Yes! Yay! I was distracted by the toy and you were answering my question. Grown-up moment. Yeah, they break down proteins. So proteo means protein. Lytic is a reference to lysis, which is the breakdown or the splitting of something. So proteolytic enzymes that break down, meaning cut into pieces, other proteins. So it's a dangerous thing to be associated with a caspase because it's going to break down proteins. Okay, and so the initiator caspase, which is initially inactive, is called a procaspase. When it becomes active, it's called a caspase. So this is a naming trend that's sometimes used in biological circles to describe the before and after. So procaspases, which are inactive, meaning they're not breaking down other proteins, or a word that people use associated with breaking down, I'll put it up here in parentheses, is cleave. That's a common word in biology, like a meat cleaver is a big knife that cuts meat. To cleave proteins or to cleave nucleic acids, that's often the verb that people use. So procaspases are inactive. When they become activated, they are then called just caspases. So they lose the pro. All right, so these caspase exist in your cell. They're hanging around. They're not bothering anything or doing anything because they're procaspases. Then a ligand comes along, binds to a death receptor. Then two other death receptors come along, and now you've got this little trio up to no good. And that trio associates with an initiator procaspase. That initiator procaspase, when it comes together with others of its kind, gets activated, and now it becomes a caspase. In a dark, ironic, I guess biology can be ironic. The reason that these caspases become activated, you can look at the difference between these two drawings. I know these are not biologically accurate drawings, they just look like blue chicken nuggets. But if you notice, this section of the procaspase is missing down here. So that's actually a reference to the fact that procaspases get activated because a piece gets cut off. So ironically, the procaspase 8s activate each other by cleaving each other, and then they become active. So they live and die by the same rules, I guess you might say. All right, so what happens to the initiator caspase? Well, the initiator caspase is not going to destroy the cell, but it is definitely playing a role. It's like the behind-the-scenes collaborator, the accomplice. So where are we at? 2. So we need 3. So number 3, now that we have a little sidebar about caspases, the third thing that's going to happen is that the initiator caspases, and since I'm not saying procaspases, it's implied that they are active. So the initiator caspases cleave and activate the executioner procaspases. Sometimes people call these effector caspases. If you take a different course, you might hear somebody call these effector caspases instead of executioner. I'll follow your book's lead since that's the diagram that you have access to. But it's the same thing if you see it worded differently somewhere else. So the executioner caspases are now active, having removed their inhibitory component. They are now ready to do their work. And a diagram, I don't think I include a diagram of this. Do I have a big diagram? No, I don't. The caspases, both executioner and initiator, have real names. So the initiator caspases, you don't need to know this or write this down. I'm just telling you, it's not this vague in real life. These guys go by the names of caspase 8, caspase 10, caspase 12. The executioners are caspase 3, 6, 7, and 9. I think caspase 1 is an initiator caspase, if I remember. But there are a lot of different ones that fall into these two categories. So it's not just two different enzymes carrying out this work. It's an elaborate web of death. Title of my next novel, Elaborate Web of Death. The caspase story. All right, so what happens when the executioner caspases are activated? So we're now at number 4. And in number 4, the executioner caspase cleaves cellular proteins such as actin filaments, thereby causing the cell to shrink and eventually form blebs, one of my favorite words. So let's say that a different way. The executioner caspases cleave and destroy what would you call a component that you have to have? Critical. Cleave and destroy critical cellular components and so the cell is starting to break down from the inside. All right, now when a cell would die a necrotic cell death, so let's say that here's one of your cells, everything's going fine, and then something terrible happens, and the cell membrane bursts, and all these things that are inside the cell leak out. Okay, when that happens, your immune system senses that. You have white blood cells circulating through your body, and they sense something bad has happened. They sense molecules that are out in the bloodstream or out in the tissue that are supposed to be inside a cell, right? It's not normal. And so they initiate an inflammatory response, which means they send white blood cells to the site. That site gets angry and red and swollen, and cells come in that are designed to destroy other cells and destroy viruses and pathogens. It's a mess. And that's fine, because if something bad happens to your cell, your body should respond appropriately. But sometimes that's not a good thing. Sometimes you don't want an inflammatory response if a cell just has to die. If a cell is pre-tumorigenic and has mutations and something needs to be done to take care of it, you don't need your immune system to have a 3-engine fire alarm and go nuts. You just need to kill that one cell. That's when apoptosis is appropriate. Apoptosis is also appropriate in places where an inflammatory response would do more harm than good. For example, if you have an eye injury, you don't want your eye to swell up and turn puffy and red and fill with liquid, right? Then your eye won't work. So some locations in your body, like your eye, for example, are called immune privileged sites, which means that they don't have an immune response normally. Like the inside of your eye does not normally have an immune response. You don't have white blood cells inside your eyeball. It would be dangerous to yourself. And so sometimes this is something you don't want to happen. Okay, that's when apoptosis really does its work. So if we want to avoid that, this is leading to something, I promise. We want our membranes to bleb. Okay, so we cleave and destroy critical cellular components. Cellular membrane now starts to bleb. This is a verb, believe it or not. Membrane blebbing is a thing. And what that means is small membrane-bound pieces leave the cell. So truly, there's not a better word than blebbing, because it looks like this. Little pieces of the cell start to just fall off. The cell isn't bursting open because this would trigger the immune response. Instead, the cell is having this incognito death where it just packages up little pieces of itself and sends them out in these membrane-bound blebs. Okay? I don't know why I did quotes. They actually are called blebs. And so the last page of the story is that these little membrane-bound blebs are trying to keep the immune response at bay. They're trying to avoid the cellular response going nuts. And so what happens then, nearby cells phagocytose and destroy the blebs. Meaning that other cells in the vicinity are going to gobble up those little cellular pieces and just digest them. They're going to eat the cell. Long live phagocytosis. This is happening right now as we speak. If you are really in tune with your body, you might be able to feel your cells blebbing right now. I want to show you something if I have access to the web. Do I? Ooh, I do. Let me read this story real quick. That's okay. I'll do it later. Cells. Oh, there it is. View feature offers new. Reject everything. Does anyone see a tab that says resources? There it is. I want you to see what. Nope. By disease area. Anyone see apoptosis? No. Cell death. Regulation of apoptosis. All right, so this is what's really happening. I don't want you to think we've learned the whole story yet. It's a little more complicated than I let on. And so here is an example of the 3-part death receptor. There's our little ligand. When that receptor is bound, it activates the association of these adapter proteins, which then recruit proteins like the initiator caspases 8 and 10. 8 and 10 then activate or alter proteins called BID. BID interacts with the mitochondrial membrane and forms a hole in the membrane. And then all this stuff starts coming out of the membrane. When that stuff starts coming out of the membrane, it activates the executioner or effector caspases 3, 6, and 7, and 9. And those start to break apart things that will then lead to cell death. So there are a few more players involved than your diagram lets on. It's a beautiful thing how complicated it is. So just memorize all of this by Tuesday. You'll be fine. If you're a literal person, I'm joking. Don't do that. Okay, so that's cell death in a nutshell, for lack of a better term. And now that we have seen cell death, let's now try to be a little more positive and look at cell cycle. Or wait,
Well,
do I want to do cell cycle or is it mitosis? Let me see, hold on. The words are sometimes used interchangeably, but this is,
Mine of mitosis is just like a chunk of...
yeah, that's what we want to do. Yeah, so mitosis is a part of the cell cycle, but only a part. So let's take a look at that. Let's take a moment. We've photosynthesized, we've died. I just need a minute. I haven't had Diet Mountain Dew in over 2 weeks, so I'm kind of crashing right now. Junkie returning to my drug of choice.
Was it worth it?
Oh, it was worth it. It's never tasted better than after being away for 2 weeks. My old friend.
You could have had it every day during those 2 weeks though.
But then it wouldn't have been as good. And I'm healthier for it. Until I backpedal. All right, let's clear some board space. Anyone have questions about cell death? Anybody's soul broken at this point in the semester? I understand. Good news, warm weather is coming and it starts in this room. This is a preview of things to come. But at least outside there'll be flowers. I guess I could bring some flowers to class.
You're
Well-
wearing flowers.
Huh?
You're wearing flowers.
I am wearing flowers. Do you notice what else I'm wearing?
Star Wars.
Star Wars characters are built into the flowers. A few years ago in this classroom, up in the upper chairs, it really gets hot. I mean, it's a good 2 degrees hotter every step in this classroom that you take towards the back. And I look back in the midst of teaching and I see a guy just sprawled out. He had a Hawaiian shirt on like me, only completely unbuttoned. He was just letting his flag fly. And I thought, well, you know, wish I could do it too, but I probably should keep my clothes on while I teach. Just professional advice if any of you go into the career.
I had class in here last semester, like middle, like grade summer, and me and my friend were almost in the back door. Like we had
Yes.
2 fans on almost all the
It's
time.
rough. There's no ventilation here. The alternative is we could be freezing, so I don't know which is better. They're both unpleasant to a degree. At least if it was cold, we could start a little fire. All right, so let's talk about the cell cycle and what that means. This chapter the goal of this chapter is 2 things. 1, to understand why your cell is dividing in the first place. 2, to understand how it's dividing. 3, to understand the division of the nucleus, which is what mitosis and meiosis are. That's when you divide up the chromosomes to produce the appropriate number of genetic elements in the next generation of cells. So mitosis and meiosis are part of the cell cycle, but they're just a small part. They're just the part where the chromosomes get separated, okay? And then, what was the last part? There was another one. how that process is regulated, what proteins cause the cell to do these things. Okay, so I want to start with this concept of karyotypes. I think this is on my list. Let's see. Karyotypes and how they are performed. So this is the first topic under mitosis and meiosis. And this type of scientist that you see there would be called a cytologist, or perhaps if they were a medical doctor, they might be called a pathologist. So these are the fields. Cytology is the field of science strictly studying cells. So a cytologist would be a professional scientist whose job was to study cells, most likely to diagnose disease. A pathologist is a medical doctor that most likely, at least some of us have had not direct interactions with, but if you've ever had a biopsy done of something, if you've ever had an unusual result in your blood test, if you've ever had an unusual, I don't know what it could be, a mole removed that was suspicious, or whatever it could be. Anything that involves a tissue specimen, that tissue specimen goes to a pathologist, and the pathologist is going to analyze it for different things. And this has changed a lot over time. Now the molecular tools that a pathologist have are so precise that they can diagnose a lot of things that before required just a lot of intuition and hard work. But now it's really become streamlined. But a cytologist or pathologist might be one that would perform this technique related to cell division that we're going to talk about, called karyotype analysis. It's possible, it seems probably pretty likely that at least a few people have had a karyotype analysis in this class. You may not have even known you had it. It may have happened to you prenatally before you were even born. But a karyotype analysis is a visual examination of chromosome number and structure. Okay, so if there was some suspicion prenatally that a fetus was developing abnormally and that there might be an issue with the number of chromosomes they have, if there was a question about whether, what's another good example? If there was a question about whether some white blood cells that looked abnormal in a blood test were precancerous, like leukemia cells, for example, you might do a karyotype analysis. If you were in the childbearing age and you were unable to conceive and had been trying to conceive, or if you'd had a number of miscarriages or at least a miscarriage and you wanted to know what caused that miscarriage, oftentimes is not a strong enough word. The majority of times, miscarriages occur because the fetus or the embryo, depending on the stage, has an abnormal number of chromosomes. And so that can be determined by karyotype analysis. Now, most people probably wouldn't analyze the first miscarriage that happens, but if you have chronic miscarriages, then usually people want to know, is there something we need to do to prevent this from happening again? But the other side of that, the corollary to that, is if you were trying to produce offspring and fertility had been unsuccessful, then you might have a karyotype analysis, because a common cause of infertility is an abnormal chromosome number. And that abnormal chromosome number might not affect you in many other dramatic ways. You might feel totally normal otherwise, but be carrying an extra X chromosome or an extra Y chromosome or only one X chromosome. You'd probably know that, but maybe not. It just depends on the circumstances and on your medical interactions. And so karyotype analysis can help a lot of people at a lot of times in life to figure out if something is happening in a cell that has to do with chromosomes that is outside the realm of what we would loosely consider normal, a term we have to use with a grain of salt, of course. What does normal mean anymore? But karyotype analysis is done in a stepwise fashion. And so what we're trying to achieve with a karyotype analysis is a visual assessment of chromosomes. So this, for an example, is an old version of a karyotype analysis. I thought I had the newer version. Oh, there's a newer version. Newer versions usually have fluorescent dyes associated with them. But either one of these would be an example of a karyotype. And so if we had this in front of us, we could count the chromosomes. Whoever put this one together, whatever pathologists or cytologists put this together, they arranged them by number and by size. And so we could then say, okay, everything looks okay, or hey, they're missing a chromosome, an X chromosome, or they've got 3 chromosome 21s or something like that. So the first step to doing this is to obtain a cellular sample capable of undergoing cell division. So this means the cells that you are looking at have to be able to divide or this process won't work. Okay, so an example, the most common example would be just white blood cells, right? If we take a blood sample and remove the red blood cells, which do not have nuclei and are not going to help us understand chromosomes, if we take those out of the equation and we just have white blood cells left, that's a good example of cells capable of division. Bone marrow cells, good example, capable of division. Fetal cells are dividing like crazy, good example that we could use to analyze them. So I'm just going to put blood as a good example. Okay, number 2. We want those cells to then undergo cell division. We want them to undergo mitosis. All right, meiosis is for cells that are going to produce sperm or eggs, and we don't need to worry about that at this stage. We just need to count the chromosomes. So we're going to stimulate the cells to divide by treatment with a mitogen. An expression I learned when my kids were in elementary school was use context clues. I'd never been taught that expression as an old timer. Hold on, I need another hit. Student said, Dr. Payton, are you going to get jacked up on Mountain Dew? I didn't understand it was a Ricky Bobby Talladega Nights reference at the time. Just thought, I don't think so. Hope not. Got a job to do. Yeah. There's no time to get jacked up on Mountain Dew. But to get back to that, using context clues, if you were taking a standardized test and you did not know what a mitogen was, but you read the sentence, stimulate cells to divide by treatment with a mitogen, and you had to guess, what would you guess a mitogen does to cells? To? Keep going, you're right.
To divide?
Yeah, it stimulates them to divide. It does exactly what the sentence says. And so you could ask, what is a mitogen? And it's something that stimulates cells to divide. There are lots of different mitogens. Growth factors are mitogens, some hormones are mitogens, some proteins bacteria produce are mitogens, viruses are mitogens, they stimulate cells to divide. So that's what it means. There are a lot of different chemicals a person might use, but they're stimulating cells to divide by treatment with a mitogen, and that is simply a substance that causes cells to divide. So, good use of context clues. Always use all the resources you have available with you to succeed. All right, so number 3 is where we're at. All right, so we've stimulated our cells to divide. So now we've taken some cells out of our patient, or out of our pregnant mom in some cases, or our cancer patient, or our fertility clinic patient, whatever it is we're trying to resolve an issue with, and we've stimulated those cells to divide. So keep in mind, what we're talking about when we say these cells is that we have cells in a dish in a lab. So these cells taken from a body can actually live in media in a laboratory. Just like bacteria can grow on an agar dish, or yeast can grow on an agar dish, you can put cells from a human or any organism into liquid media, and if you give them the right nutrients, they'll survive, at least for a time. They won't live forever unless they're cancer cells, then sometimes they will. But they'll be able to survive. So these are sitting in your lab, and you're stimulating them with the mitogen, and when you do that, they start to undergo cell division. Okay, so you're on your way. Now, you've treated these cells up to this point as though you wanted them to undergo division, and now you're going to change courses. You are going to initiate cell cycle arrest, which means that division stops, just like it says, context clues. So if somebody says that cell cycle went into arrest, it means that it was dividing and then boom, stopped. So you're going to initiate cell cycle arrest by treatment with a chemical called colchicine. Colchicine is a drug from a plant called the autumn crocus, which is a shrub. As a general rule, don't eat shrubs. A lot of them contain toxic chemicals because a shrub can't run. So in order to protect itself from being eaten or being predation being played upon it by other organisms, sometimes they produce toxins. One of those toxins produced by the autumn crocus, a sweet little plant otherwise, besides its poisonous nature, is colchicine. And so colchicine works its magic by causing cell cycle arrest. Okay, and colchicine does this even though we haven't talked about mitosis yet. You may remember in mitosis, I bet you remember, that at one point, well, we talked about cell anatomy and brought this up. At one point, microtubules reach out and grab onto the chromosomes, right? And so you see lots of pictures where chromosomes are being grabbed onto by microtubules. And so what colchicine does is it blocks microtubules and pauses cell cycle at metaphase. Now there's a reason that it pauses the cell cycle at metaphase. Well, the reason is it blocks the microtubules, but there's a reason it's desirable to do this. And the reason it's desirable to block the cell cycle at metaphase is that this is when the cells are the chromosomes are most visible, right? If we just took the white blood cells directly from the blood and put them under a microscope and looked at that nucleus, it would just look like that. We wouldn't be able to tell anything. But if we take this white blood cell and we let it go to metaphase and we look at those chromosomes now, they're going to be like this, right? Getting ready to line up at that metaphase plate. They'll be very visible. So that's the reason that we've teased the cell into going into the cell cycle, and then just as it was having its time dividing, we zap it with colchicine and it stops. And all of the cells in your little living system here are now locked into metaphase. Okay, different use of the expression locked in. The original use of the expression. And so what do we do now? We have all these cells that are in cell cycle arrest, and so now we are going to stain the chromosomes, lyse the cells, and count and analyze the chromosomes. All right? DNA normally is not visible until we stain it. Oops, sorry. Shields down. So we have to do something to make the DNA visible. So you can stain it with a dye that causes bands to form. Okay, they're called G bands because the dye is spelled with a G, Giemsa. Or you can stain it with a dye that causes the different chromosomes to fluoresce a different color. That's really super convenient. Whoever conceived and invented this process, changed karyotype analysis forever. Makes it so much easier to count. But if you were old school, you'd be able to look at the pattern of bands on a chromosome and say, that's chromosome 16, which is amazing. But once you can visualize them, then you can see, again, the number, the size, whether a piece might be cut off of a chromosome, that can happen, and that leads to some pathological issues depending on the chromosome. Lysing the cells, again, this process has lots of variations. You can lyse the cells. You all being specialists in osmosis know that you could lyse cells by creating an unfavorable osmotic environment, right? You can create a really hypotonic environment and shrink the cells, hypotonic would cause them to swell, not shrink. And so if you can cause the cells to swell, they can burst. You can also use chemicals that cause the membranes to break. A way that people used to do it is they would put their slide that they're going to analyze their chromosomes on the ground. They would put their cell sample in a medicine dropper and they would drop it from here and then splat. When it hit the slide, dropping 5 feet or so, it would just burst and the cell would break, the chromosomes would spread out. And they don't look like this when the karyotype is almost done. They look like this. So the job of this technician or cytologist or pathologist, whatever they might be, is to arrange this into something usable. In fact, you can see they've taken a photograph. So in the olden days, they would take a photograph and cut out each chromosome and then create a diagram like this where they pasted on their cutouts in order. In today's technology, of course, if you just had a Snapchat photo of that screen and I gave you 18 seconds, you could arrange these into a new photograph altogether just by manipulating it with your pinky finger. And so it's a different world, certainly. Digital technology has changed medicine in many favorable ways, for sure, and that would certainly be one of them. Okay, how do you feel about that? Feel analyzed? You may have a karyotype done someday. Now, when the doctor does that, you could say, hey, I know what's going on. Care if I have a look? Or you can just say, please let me know the results. Okay, now, we've got a few minutes left and we better take advantage of it so we don't get behind, make sure we have plenty of time to review. Let's just dip our toes into this topic and then we'll call it a day here in a few minutes. So the cell cycle is the process we see up here. This looks more complicated than it needs to be, but the cell cycle revolves around, and let's see, your book does it at the bottom. I do it a different way, but I'll follow your book's lead here. I think of cell cycle as being kind of like the face of a clock, right? Where we are moving in a clockwise direction. Fun fact, this is one of the only rooms in this building that has a working clock. They used to have clocks that were all synchronized. So every clock was right all the time. And one of the tricks during daylight savings, when the clocks had to be set back an hour, how would you set a clock back an hour if you were in a classroom and you needed this to be 9:40 instead of 10:40? What would you do? Yes, you would take the clock down and set it. But the solution at the time, this was a previous building staff, so I'm not being critical of our current building staff. They're great. But at the time, what they did was they turned the power off for 23 hours to the circuit that fed the clocks and then turned it back on, unless they forgot. And so the next day, when people came in, none of the projectors worked because they were all on the same circuit. And so we couldn't turn the projectors on, and everyone was like, what's going on? And then the clocks, of course, aren't ticking. And they're like, oh, serious? That's the solution? A good idea in principle, except lots of things are running on those circuits. Okay. But I'm glad we have some captains of industry in here that are able to do good solutions. That's all it would have taken. 2 seconds. All right, so let's define this and then we'll call it. So the cell cycle works in a clockwise fashion if we diagram it like this. And the other thing to consider with the cell cycle is that it only goes in one direction. This is a really important concept. Did I turn my thing off? It's not me bleeping, is it? No. This is a really important concept because you can't go backwards in the cell cycle. And so for these 4 stages, which we'll define, once we're in a stage, that's it. We can only move forward. We cannot go back and undo everything. And so let's define these stages and then we'll make that our final note for today. The first one is the G1 gap phase. Okay, and leave some space here. If you're taking notes, we're gonna have about 2 to 3 sentences to say about that. And then the second one is the synthesis phase. Leave a little gap for a couple sentences. The third phase is the G2 gap phase. The fourth phase is the M phase, which is, depending on the type of cell we're talking about, either mitosis or meiosis. I think in this diagram they just have mitosis. And then you can leave a little gap there. And then there's one phase that isn't, I don't think it's depicted on there. No, it's not. But we don't want to forget about it. And on Thursday, make sure I don't forget about it. We'll pick up here. It's called the G0 phase. And the G0 phase is kind of off of our clock face. It's out to the side. It's an obnoxiously large 0. Should be just a subscript. Okay, so let's call it there. It's hot. Go get a cold drink somewhere. Rehydrate.